got his PhD and postdoc in neuroscience at the University of California, San Diego. Dr. Haag is currently professor and chair of the Department of Biomedical Sciences here at ETSU. He has interests in neurotrophic factors, spinal cord injury, and neurogenesis stroke fields. Dr. Haag?
Well, thank you for the invite, Dr. Goodkin. I feel like I'm being examined by an austere panel of psychiatrists, but as a professional, which is better than as a patient. Anyway, I'm delighted to be here. If it's too loud, too soft, tell me. And I think there's going to be more folks coming in. And what I'll do today is talk to you about a subject that many of you have heard about, and maybe some of you are interested in. I know at least one person in the room is. But it doesn't come up very often in regular medicine. But this is a field that's developing. It's fairly new in the broad history of medicine, and it's probably going to have an impact at some point, which means we're very early days in understanding how new neurons, this neurogenesis, how that is occurring in humans, healthy humans, and ones with disease. What I'll show you today is all animal work and some tissue culture work, and point you in the direction that the field is thinking that it will have an implication for human therapies. Also, we work on neurogenesis because you can make very pretty pictures, as these ones. Just a disclosure, I have no disclosures. I'm very poor. Anyway, this is the team. This is what makes everything possible. So everything you see is because of these folks. And I get the joy of mentoring and guiding them and interacting with them on a daily basis. So the outline for today is I'll talk about neurogenesis, what that is a little bit, how it has maybe implications for human disease, and how neurogenesis in adults is regulated, and what is known about the mechanisms, the molecular mechanisms that do that. There's a couple pathways that I'll talk about, which are transmitters, and I'll talk about things that are used in medicine, and then some of the discoveries that we've made in the last few years. And also something that has really excited our lab is some almost serendipitous finding, which after, I'm not going to say how many decades, but finally is getting us into doing and planning some clinical studies. Throughout, I want you to think about, and I'll show you some directions we're thinking about, how can you treat people ultimately with pharmacological agents. So neurogenesis, before 1990-ish, in the general field of neurosciences, was thought to not occur in the adult brain. Our grandfather in the neurosciences, Santiago Ramon y Cajal, said, once you're done, you're done. Remember what you learned in high school. You need to preserve your neurons because once you lose them, you don't get them back. Well, that changed quite dramatically, and I was fortunate to be at the front end of the discoveries of neurogenesis in the brain. So the late 90s, it was discovered that in the hippocampal formation there is neurogenesis occurring, and then this region here in rodents that goes to olfactory bulb. And currently, there are several other areas that have been discovered where there are stem cells that proliferate and that produce new neurons. Now, in rodents, and here you see a mouse brain on the side, and these two regions, it's not a lot. So here we see cells that have been marked with a proliferation marker. There's not thousands of cells in this region. In a mouse, maybe there are per day. Yes, 1,000 cells made. They go to the olfactory bulb. You think, well, they're going to have a big nose at the end of their life, but it's compared to the millions of cells in their brains, it's a small contribution over time. In humans, it's even less spectacular. But I'll get back to that in a second. So there are different markers that we can use. For instance, these proliferation markers, we can label them, the cells with BRDU that incorporates into the DNA. So if a cell proliferates, it gets this label. And then surrounding them are these red things here. These are astrocytes. And these are sort of our popular cell type. We want to focus on those. We don't want to focus on neurons. Neurons die or degenerate in many conditions. Astrocytes are the ones that we want to focus on. They tend to survive and to help the brain. So just a 101 for neurogenesis. So there are neural stem cells, just as you have stem cells in every type of tissue. These can make astrocytes, oligodendrocytes, and neurons. And they tend to do this under normal conditions. They tend to make only neurons. So here we've got the stem cell. It self-renews. And then once in a while, it produces a daughter cell. And those progenitors then go on to proliferate. And this is an important aspect of our studies where do you start promoting more neuron formation if you want that. The other thing that you notice here is that the different cell types, these B1s, those are actual astrocytes. So astrocytes can actually convert into stem cells and then start this process of neurogenesis. So in the humans, now just as a comparison, of course humans are much bigger than mice, right? So here's a brain of a human, and here's a little mouse. It's much, much smaller. So if you think of either psychiatric disease or acute trauma, and we want to think about cell replacement, the human brain is much bigger, many more cells, much farther distance to travel for new cells to reach their target. I hope you can see it, that here we've got a cross-section of a human brain, and there's just little areas around the ventricles. And in the mouse, they go to the olfactory bulb. In the human, they go into the striatum, caudate. And it's thought that there's turnover in the caudate in our normal lifespan. And that these neurons from this, it's called the subventricular zone, under the ventricle lining, that those are replacing those cells on a constant basis. The same may be true for the hippocampal formation. So here you see, hopefully, you can see that there's the hippocampal formation in the dentate gyrus. There are cells that proliferate and add to the dentate gyrus neurons. Lots of neurogenesis in the subventricular zone and less so in the hippocampal formation. So most of our studies are in the subventricular zone because we want lots of and fast neurogenesis to happen so we can manipulate that easier. Now, in terms of disease, these are kind of the things that the field is thinking about that neurogenesis is involved in, and where we might utilize or capitalize on the existence of neural stem cells in regions that are close to it. Now, I don't have on here normal aging, because it's not a disease, although there are many days that I probably think, but during normal aging, we're losing the capacity to make new neurons. And in the hippocampal formation, of course, it's important for certain types of memory. And so we want to perhaps, during regular aging, when we have cognitive decline, want to understand whether neurogenesis has a role, or decline in neurogenesis has a role in that. So one can imagine that in Huntington's disease, where the caudate is affected, the subventricular zone that just sits next to it could actually start making neurons, more neurons, that if you had a drug that could induce that and give you more new neurons, that might be helpful. The fact is, we have no clue whether that's going to work, right? So we're basic scientists. The field of neurogenesis is so early that we can't predict whether or not it's going to be useful. Now, in animal studies, there seems to be a lot of promising findings. And that's true, too, for mood disorders. Van Gogh, of course, had all kinds of issues. Alzheimer's disease, in the hippocampal formation of Alzheimer's patients, there's a decline, a drastic decline in neurogenesis. The beta amyloid is toxic to neurogenesis. If the NIH is kind to us, we will study this in the next couple of years if we get a grant from them on Alzheimer's in Alzheimer models, mouse models. So there is some hope that this process could be utilized for repairing or reviving areas in the brain that may need that. Now, I'll confess, if I see an image like this, I'm going to say, that's a tough job, right? How are you going to replace all of that? And I remind myself always that, well, I'm here, I have a brain, and that came from one cell, the original stem cell. So it is, theoretically, it's possible. Whether it'll happen in my lifetime, I doubt it. Anyway, so for those who are into treating people with eating disorders, the hypothalamus, of course, is involved in that. And probably a decade ago, it was discovered that also there was neurogenesis going on, and that it was regulated by a molecule that's called CNTF, and I'll get back to that in a second. So yes, there's a lot of areas that we can focus on. Now, how are you going to treat somebody in the future, or a mouse, or a rat, to get more cells? Well, you could put a whole bunch of cells in the brain. If you think of a stroke, and you put millions of cells there, maybe this will help. The problem with that is, if you put a cell in a toxic environment, in a degenerative environment, they also are affected by the milieu that causes their cell loss. So you can think of injecting growth factors. Our approach is pharmacological drugs. It's easier to titrate. It's easier to stop. It's easier to give intermittently. If a new drug comes on board, you use that one rather than being stuck with whatever you put into the brain as let's say, with a virus expression system. Exercise is known, running is known to induce neurogenesis. Intermittent fasting or caloric restriction is known in animals, known to induce neurogenesis. And then most recently, because everybody's talking about microbiome, right? It influences everything in your body. It's important for cancer treatments. Just a paper in Science or Nature that shows that immune therapy of cancer is very much influenced by your gut flora. And so also in the neurogenesis field, this has become a topic of interest in that if you have antibiotics, it reduces neurogenesis in animals at least. If you have probiotics, think of your yogurt, eat your yogurt, you're going to be smarter. All right, so I'm interested in CNTF, ciliary neurotrophic factor. For those who are not familiar with that, if you think of nerve growth factor, I hope you're familiar with that. That was one of the first trophic factors that if you sprinkle them on neurons, these neurons love it and they survive and they grow axons. The second factor was ciliary neurotrophic factor. I was privileged to be in the lab that discovered it. I might look a little different at this point, but that is what started my career at UCSD, and I've stuck with this molecule. And I'll tell you in a second why. So it's a member of the cytokine family that includes interleukin-6, which probably all of you know is a pro-inflammatory cytokine. And then the other one is LIF, which by itself is also involved in neurogenesis. So back then, I was interested in, and still am, in rescuing spinal cord or the brain against acute insults, like spinal cord injury, stroke, etc. And had shown in a number of models, for instance, in Parkinson's models, that we could rescue the neurons against degeneration. At some point, we discovered that motor neurons are also rescuable with CNTF. So large clinical trials, phase 2 clinical trials were started with injecting CNTF in patients, ALS patients. And it failed miserably. There were 1,300 patients. And it really put a damper on the field of neurotrophic factors. There was very serious side effects, cachexia, coughing, all kinds of side effects that stopped that trial. And nothing was gained because the CNTF did not get into the nervous system. And at some point I thought, well, don't feel sorry for yourself. Think how you can solve it. So that's how we came up with the idea of, well, wait a minute, if astrocytes, which make CNTF in the brain, in the spinal cord, if we can coax them with a drug, with a pill, to make more CNTF, would this be good? Could we circumvent the low bioavailability in the CNS? The other thing is that CNTF is almost exclusively only made in the nervous system. A lot in the Schwann cells of the periphery. But if it's only made in the nervous system and we pop a pill that has no side effects, then we circumvent the side effects of the CNTF. So that's what started a long history of things in the lab that we've pursued. So CNTF, you need to link it to neurogenesis. That's not necessarily a very clever thing that I thought about. But at the time, because I'm working on CNTF and the neurogenesis field started, we said, well, wait a minute. Why don't I just see whether CNTF has a role in neurogenesis? And lo and behold, it did. And that has started a long program in the lab that we tried to increase CNTF expression. So here you've got in a mouse, here's this little triangle next to the lateral ventricle that has labeled cells that were labeled with beard use, they're proliferating. And here you've got antibodies to CNTF that were injected here in the ventricles, and you stop that proliferation, or you reduce it quite a bit. If you give, in contrast, give CNTF, you can get more. And this is just quantified here. For instance, here's antibodies, so you get reduction. At some point, people made CNTF knockout mice, so they have a mutation where they don't express the CNTF protein. And if you compare, here's a saline-treated wild-type mouse with a number of these cells, and in the knockout, there's fewer of them. So this suggested to us that CNTF regulates, promotes neurogenesis. The other thing I'll get back to in a second, you see Quinn here, can increase it. This is a drug. So that was a good thing for us. So where do you start? Because at the time, so okay, there's all kinds of growth factors that were starting to be known to induce neurogenesis. And how do you get to a pharmacological drug to actually be able to do the same thing, to induce CNTF and then induce neurogenesis? So if you have friends and you read their papers, and remember the papers, then you might get ahead. These days you can just do PubMed. Back then we did not have PubMed necessarily. You went to current content. You remember that. So John Rudge had shown that if you take astrocytes, cultured astrocytes, you treat them with forskolin, which is a cyclic AMP inducer, then CNTF went down. So I thought, wait a minute, could we do the reverse and reduce cyclic AMP and get an increase in CNTF? Well, okay, you could do that, but of course cyclic AMP is very important for all kinds of processes in cells. So you got to get more specific, and with just to cut to the chase, we found that D2 dopamine receptors, they're GI-linked receptors, so they're going to reduce cyclic AMP. And lo and behold, they're expressed in astrocytes. This is an astrocyte in that subventricular zone. And said, okay, let's try those. The other observation we had, which helped with the picking dopamine receptors as a target, is that when you look at where CNTF, and I'm afraid this is maybe not going to show up very much, these are reporter. There's a reporter in the gene that you knock out. So wherever it's normally expressed, you'll see the reporter. So there's not a lot in, if you stain for CNTF, I might be able to show you that in a second. There's a lot more. But it's in this rim here. And the neurogenesis also, just next to the lateral ventricle, this is the striatum. So in a mouse and a rat, the striatum is caudate putamen together. So you can see that it's limited here. But wherever you see, in this case, tyrosine hydroxylase, or dopaminergic projections from the substantia nigra, and of course, here, ventral tegmentum. So wherever you see neurogenesis, we saw dopaminergic projections. So we put two and two together, and this slide shows you a series of studies led by Pang Yang and another person who's not on here, Sarah Baker. And we're talking about ancient history. I'm sorry to go through that, but it's, I think, relevant to what we're talking about today. So we did a very large series of studies to suggest and propose that from the substantia nigra, Parkinson's disease, right? There are projections to the subventricular zone. There are also projections to the hippocampal formation from substantia nigra. Not a lot, very few, but they're functionally important. And through D2 receptors, there's induction of CNTF expression in the astrocytes, which then regulates and promotes neurogenesis. And I'll take you through some of those slides. So here we've got saline injected into mice systemically. Remember, I want to discover a pill that you can pop and be happy all the time. Okay, those exist through CNTF. That you're healthy because you can make more CNTF in your brain. So we want to treat people systemically. So here's a mouse, for instance, that has quinperol as a D2 agonist. It has D3, for those who study it and know it and use it. It has some D3 activity. In subsequent studies, we've shown that the D3 receptor is not involved in neurogenesis. So we're talking about D2 type of receptors. So quinperol, if you give it, it increases CNTF. Here are just other types of mice. We got two types of backgrounds. And here again, quinperol in wild types, and then of course heterozygous here. So it induces CNTF. You can see here that the BRDU, that label that we inject to label proliferating cells, that there are more cells. Now we're talking about treating a mouse, or rats for that matter, for 2 days with quinperol. And they've got a substantial increase in neurogenesis. That doesn't happen in the knockout, the CNTF knockout mouse. So it suggests that indeed that quinprol through D2 receptors activates neurogenesis by making more CNTF. What's not on here is if we look at Parkinson models, we can see that there's a decrease in neurogenesis. This is also true for patients, or Parkinson patients have less neurogenesis in this particular region. Here you see a slide. It's yellow because red and green together makes yellow. Red is CNTF, yellow is astrocyte. So there's a lot of CNTF in that region where you have neurogenesis. All right. So then we thought, why do we have neurogenesis only in that thin rim around the ventricles? But you've got dopamine everywhere. And so just speculating that maybe 5-HT receptors, which at that point, serotonin receptors, became known in the field, the stem cell field, to induce neurogenesis. And I'll get to that in a second. The projections that you see, the fibers, as you can see here, but also the receptors are seen in that little region. So it made sense to say, okay, dopamine plus 5-HT overlap, this is driving specificity for CNTF expression in neurogenesis. We were wrong. In our hands, 5-HT agonists for the 1A receptor, so think 8-hydroxydipad, for instance. It didn't work. It does not work in our hands. Now, this is a problem. You can see we're 2012. We're up against a publication that changed the field of depression, at least in the thinking of how neurogenesis fits in that. That if you take 5-HT1A receptor knockout mice, you give them fluoxetine, like here, or D-pad, which is 8-hydroxydipad, you don't get more neurogenesis, which you see in the wild type, and you don't get the behavioral benefits, for instance, of fluoxetine in depression models. So we did talk to, some of you may know, this person, Renee Han, who's very famous in the neurogenesis and depression field. What seems to be the case is that we used naive mice, and so we could not push their neurogenesis beyond a certain limit, whereas Dr. Han and their group used a depression model which decreases neurogenesis. And they could reverse that with drugs that you're all familiar with. So you got to work on things that are exciting and positive, and they get you grants, all right? So we said, bye-bye. We need new things. And this is where, again, for those who are into research, if you're going to be physicians and treating patients only, keep your eye on the science. At least read the summaries. So I remembered, wait a minute, if you poke a hole, when I was a younger man, you poke a hole in the brain, it has lots of CNTF expression. So what is regulating that? And nobody up to that point seemed to know. You're sort of waking up the astrocytes to become reactive, and the dogma or the thought is that the astrocytes are trying to help the poor neurons that need to survive, which may be actually correct. So Song Kang was a postdoc in the lab and showed that over time, and this is an MCAO, this is a stroke, and in a mouse or a rat, you can feed a little filament up the internal carotid to the MCA origin and stop that blood flow, and you get an infarct in mice. Here you can see, in rats, you can see there's loss of neurons. And lo and behold, after 1 hour, 2 hours, it just starts skyrocketing. So something about stroke And we thought, okay, is this maybe loss of neurons is inducing CNTF? So we did the next thing, which is quinolinic acid. This is an excitotoxin. It gets rid of neurons. Glia are fine. Microglia are fine. Astrocytes are fine. And it has exactly the same response in terms of CNTF. So if we compare the MCAO, which is a stroke, to quinolinic acid, you can see there's very similar levels of CNTF. There's a little bit of a time difference. But, so this told us that neurons are somehow suppressing, perhaps, CNTF expression. Now, since we're talking about stroke, just in between question for medical students, what's the first thing you do when somebody has a stroke in your family or, God forbid, here? What's the first thing? Remember what Dr. Kimbrough said. Louder? There you go. Why? Okay. Why don't you drive them to the hospital? So, and this is key for everybody here. Activate the system. Remember, if somebody has a stroke, you need to figure out, is this hemorrhagic or is it occlusive? And what do you do to distinguish the two? There you go. So remember this. If there's anything from this lecture, remember. This is very, very important. You activate the system because otherwise you come in and say, I need a CT scan. Oh, that's going to take half an hour to organize. Anyway, back to basic science. And I tell this every time I give a lecture on, and it relates to stroke, I tell this because this is going to save people's lives, save people's function, because you're actually gaining time. All right. So Matt, who's in the back there, came on board and showed that in a culture system, if you take astrocytes only, here's just presented as one fold. You take the astrocytes as the controls, and if you put neurons on top of them, boom, there goes the CNTF down. So something from neurons is actually reducing CNTF expression. When he went in and lightly took off the proteins, there was more CNTF. The short of it is that he showed that this RGDS, which is an integrin blocker, increases CNTF. It suggests that integrins, if you remember your basic science, cell biology, that they regulate and suppress CNTF. Now, there's a lot of them, and I'm not going to belabor you with this. The only thing is that they do signal, so this is a good thing. And so Matt went on, together with Chiharu, to show through a long series of studies. For instance, here you can, if you remember, all these ligands around the cells that bind to the integrins, that you can kind of distinguish classes. And through a number of studies with antibodies and blocking peptides, he and Chiharu in the lab discovered a pathway that where neurons are binding to integrins, specific integrins, and through FAK and junk and this STAT3, this pathway is blocking CNTF expression. And this started off a very interesting series of studies where Matt and the others showed that if you give here FIK14 or a PF573, which is blocking this red, the brake, then you get more neuroblasts. So here's a picture of that. Here's quantification. You get more CNTF, and you get more neuroblasts. So if you block this pathway, you're getting more neurons. The same is true for when we do systemic injections. For instance, Gia came on board, who's also here, and showed through a number of experiments. For instance, here you've got, again, that subventricular zone. In this case, the beta-gal is the reporter for CNTF. So if you give peripherally this FAK inhibitor, then you get more CNTF expression very widely. And it's quite specific because LIF and IL-6 are not being induced. If you give FAK14, this inhibitor, to wild-type mice, you're actually inducing neurogenesis, but it doesn't happen in the CNTF knockout mice. So this was good for us in defining that this pathway is very selective and selective on regulating or repressing CNTF. So we're not messing up all kinds of systems, all kinds of signaling. We're being very specific. This is consistent with current clinical trials for tumors where FAK inhibitors are being tested. And they're quite reasonably tolerated. The same is, I'm going to not go through this, but the same is for this junk, this downstream from FAK. It's important and specific for regulating CNTF, and by that, also neurogenesis. So for instance, here you can see that only CNTF is increased, not the other cytokines. And here you can see that, yes, junk inhibitor increases neurogenesis, but not in the knockout, CNTF knockout mice. So we thought, wow, this is awesome. And this is my daughter, by the way. This was her first snow. And now she has learned that the base word of snow is no. We don't like it. All right, so back to reality, which is you've so far seen a lot of things in healthy mice, naive mice, and we wanted to test whether some of these ideas are important for stroke. And this started off an interesting time in our lab where we had to learn a lot about stroke and how to do things and what other people are doing and getting funding, and all of that worked out reasonably well. So after stroke here in a mouse, just next to the subventricular zone here, you've got striatal damage, and you get infiltration of leukocytes. And when we looked at CNTF, if you give a 30-minute MCAO, so you're occluding the MCA for 30 minutes, then you get this type of injury and a big boost in CNTF. The number of BRDU positive cells, so the proliferating cells, increases in the wild-type knockout, wild-type mice, but not in the CNTF knockout. So again, it suggested to us that CNTF is a very specific target that we can regulate with drugs, but it's also very key to why neurogenesis increases after stroke. So it's a good potential therapeutic target. And we will see, maybe in my career, whether that is a true statement or not. And if it's not, that's fine too. We are having fun. So stroke induces neuroblast formation, says here. So one key thing you have to think about in the stem cell field, and you will come across this at some point, that stem cells can be bad also. So stem cells can make cancerous cells. And actually a lot of the glioblastoma field is thinking that these nasty cancers originate from stem cells. So just provoking the stem cell to make more cells is maybe not so good. So here we see that actually the number of increased cells here that proliferated is the same as the number. This DCX is a marker for neuroblasts. And this suggests to us, and other data that we have suggests to us, that all of the stroke-induced or drug-induced increase in neurogenesis makes the normal neurons. We don't get more astrocytes or more oligodendrocytes, although more oligodendrocytes would be good too, right? For which disease? Students, residents, oligodendrocytes, multiple sclerosis. So we haven't sold that as a potential target because you can only do so much in your lab. But it could be a whole grant by itself and a whole program by itself. All right, so you think, okay, this is great, right? But when we looked early after stroke, there was something funny happening. That the CNTF here in the green, this is over time after MCA occlusion. There's a lot of CNTF, but there was almost no increased neurogenesis. The short of it is we think that at the same time as you have increases in CNTF, there are counteracting cytokines like LIF and IL-6 here is much higher that block this transformation of a stem cell into a progenitor. And so we're thinking that maybe we should also study LIF and IL-6. Can we suppress them? And for those who are interested in inflammation and psychiatric disease, IL-6 is probably on your radar. So here's where serendipity came in. So you've seen this before, where we had a lot of different ligands to the integrins, and we could see that vitronectin is one of those ligands that suppresses CNTF. So one of our colleagues in Louisville said to Matt, why don't you look at LIF? And we're glad he did, because you're not often that you get results like this. So here we've got the same types of ligand panel, here for LIF, here for IL-6, and boom, only vitronectin induces these cytokines, which may reduce neurogenesis. So he went on and showed that indeed this was the case. If you take, for instance, vitronectin, and you look at IL-6 expression, and you block that integrin pathway, now you've suppressed expression of this inflammatory cytokine. Now, vitronectin is unique among these type of ligands, these extracellular matrix molecules, in that it also binds to UPAR, and I'm not going to belabor that, but it told us that we're on the right way to create some kind of specificity. Again, we want to have drugs that only mess up with certain things in the brain and regulate it. And I'm saying mess up because, of course, there are important functions for IL-6 and LIF. So we got to sort of fine-tune where we have a middle of the road type of approach. You suppress this maybe a little bit or just enough to have therapeutic benefit. I'm going to skip this because this is probably the most exciting slide that we have for today. And this is where we're getting, hopefully, into doing some clinical studies. And so here, after stroke, you can see that, and all of you know that, blood's going to leak, proteins are going to leak into the brain. In occlusive types strokes, you don't have hemorrhage. Well, you can, of course, if it liquefies. But generally speaking, we're thinking of proteins that go in there and blood products. Well, one of those is vitronectin. So every time you see plus, that's where we injected recombinant vitronectin. And it leaks into this SVZ. So we took SVZ, extracted it, and run it on here, a Western blot. You can see there's more of it. Now, when Gia and Hannah and Matt, we sat down and said, wait a minute. We know that the blood has very, very high levels of vitronectin. And a lot of it would get into the parenchyma. Would it be bad? And so they said, okay, let's go and look at plasma levels before and after stroke. And you can see that you've got high levels. We're talking about microgram per ml. This is a lot of protein in our, and we also as humans have a lot of vitronectin. That in the female, there's an increase actually as a sort of an acute phase type of response. So vitronectin is made by the liver. And of course circulates and is involved in hemostasis, all kinds of processes, cellular processes. But it didn't happen in the males. Now again, I won't get into the history of it, but this was simply a serendipitous finding. Normally we work on male mice in the neurogenesis field. We had some female mice in our breeding colonies left over. We had an experiment to do, and we did the experiment in females, and boom, there we have it. So there's a sex difference here, which we think is important. So in stroke, we can plot, for instance, individual animals. You can see every time after the stroke there's an increase. When we go in and look at cytokine expression, you can see that they're all in CNTF, LIF, IL-6, they're all increased in both male and female, but much more so in the females. When we go in and say, okay, IL-6 is bad for tissue, that's sort of a hypothesis, because inflammation is good, but it can also be too much and excessive. So when we plot out the number of the amount of mRNA or protein against how much plasma vitronectin we had in that mouse, then there's a correlation. And here are two different studies where very nicely, the more vitronectin that ran in that bloodstream, the worse, at least that's what we're saying, the higher the IL-6 levels. Now we're busy now to say, okay, doing experiments where do the high or the increased vitronectin levels in the plasma, do they predict worse infarct size? And if that is the case, we have a collaboration set up with a stroke neurologist in Louisville where we'll get stroke patients' blood, and they do all the clinical assessments and follow-ups, and we'll see whether there is some kind of correlation. The question is, and I won't get into that, can we block vitronectin's bad effects? And the answer is yes. If this were a stroke neuroprotection talk, I would get into that. So we're really excited. I told the group that if we can get drugs on board, like these FIK inhibitors, in stroke patients, in females, by the way, females have worse outcomes after stroke. That is not related to age, occurrence of stroke. I'm going to retire because I would never ever be able to surpass that. That'd be such a great gift to me personally. Here's just another little bit, and then I'll close off. But, this sex specificity of this IL-6 response is quite remarkable. So if we look at vitronectin knockout mice in the white bars, you can see that after stroke there's a large increase. It's much less in the knockout mice, but it's the same in the males. So something about being a female mouse, and we don't know whether this is true for humans, something about a female mouse makes it respond different to vitronectin than the males. And I won't go through this because, frankly, we don't understand the data. So when there's ovarectomy, this basically suggests there's an interaction between sex hormones and vitronectin's effect. And we think that's very exciting. The stroke field has tried to treat people, the neurologists, with estrogen replacement treatments. All of that has not panned out very well. And we think that we can perhaps circumvent this to focus on this FIK, the integrin pathway. Now, we're talking about neurogenesis, so yes, in knockout mice, if we look at the males, you can see that in the wild types, here we see an increase in neurogenesis, this BRDU positive cells. It also occurs after MCAO in the knockout. But in the females, this difference, percentage-wise, is much bigger. So we're thinking that in the males, vitronectin doesn't play a role in blocking or inhibiting neurogenesis after stroke, but it does so in females. Now, how does this affect you as folks who are interested in treating people with psychiatric disorders? It's quite, or degenerative disorders such as Alzheimer's that have, that you see patients. It's possible that vitronectin plays a female-specific role in those diseases also. And if you think and keep your eyes open on things like IL-6, come talk to us and we can perhaps collaborate if you have tissue samples or blood samples and you have an idea that let's figure this out. We are certainly open to interactions like that. Now I'm going to skip through this because we're getting close to finishing. So again, this is sort of the overview of what I've been talking to you about in the second part of my talk, that vitronectin through these very specific integrin receptors is activating FAK. And when we block this, then things get better. Here you just have a representation of what we think happens after stroke. We meaning the field, this blood-brain barrier breakdown and things go awry. So the short of it is, I should just skip this slide because we have not gotten the data back for FAK inhibitor-treated mice. And so in the meantime, what we've done is that, I'll skip to this, through a series of studies, Gia tested the following, which is if we block cytokine expression of these IL-6 and LIF, which acts through, it's a GP130 receptor. If you don't know it, forget it. But we put blockers in. And so if this SC144, put it in naive mice, it doesn't do anything to neurogenesis. But after stroke, you can see here the vehicle being your controls. It actually increases neurogenesis. And we think it is because we're not affecting this CNTF, which goes up in stroke, but we're reducing the effects, the negative effects of IL-6. So again, these are strategies where we're trying to find very specific targets that if there's a drug, and of course we only take targets where there are drugs, if there's a drug available or is going towards clinical trials, we favor those so that we could test them in the future. Because we don't like side effects. Nobody does. And we want to be very selective. If you think of neurogenesis and for instance, Alzheimer's disease, we probably have to treat a patient for a long time. So we need to have safe, and that means specific drugs. And that's kind of where we are. And this is the team. You can see we always smile. We may all be depressed by NIH funding, but we always smile. Any questions?
Yes, Theo, that was great. So CNTF and BDNF are both neurotrophic, yet there's a lot more research on BDNF and major depression than CNTF. And it would appear that both might be important in cooperating in neurogenesis, and both are related to countering the impact of pro-inflammatory cytokines on hippocampal atrophy, like IL-6. So I'm wondering why there isn't more research on CNTF in MDD and the interaction of CNTF and BDNF.
That's a really good question. It's basically it's related to history that very few people continued to work on CNTF because it failed in the clinical trials on ALS. So there was a disinterest in that. BDNF also, whereas CNTF expression is very low in the CNS, in the brain spinal cord, which for us is, we think is good because you crank it up. BDNF is expressed everywhere, so there's larger cellular targets. And so BDNF, the thought is that BDNF makes cells survive better. So whereas CNTF drives proliferation and formation, BDNF is necessary for the survival of the cells. So this picture here, you can see large empty spot, no neurons. Well, when some of these neuroblasts go into the striatum, they die. They can't survive there. In normal neurogenesis, BDNF is a survival factor. So yes, people have looked at BDNF in depression. We are starting to look at anxiety, stress-induced anxiety, and the role of CNTF. And the answer is, Gia, if you invite her, she will give you a seminar on that.
I was curious about something you mentioned early on. You talked about, and I know this is probably hard to answer, but I'd invite your thoughts on it, that, you know, exercise, fasting, probiotics, all of these are potential for, I guess, probiotic neurogenesis, for neurogenesis, potentially. So is the commonality, at least in terms of exercise and fasting, that there might be some sort of injury transiently that in some way, you know, you talked about how you poke the brain and you get things released. Is there some commonality or some thought you have around why or how that might be?
We have not studied that, but the answer is BDNF and serotonin. So serotonergic, this 5-HT1A receptor, and BDNF, there's a big interaction. Lots of people have studied that. So 5-HT regulates BDNF. And so running and caloric restriction, they all increase BDNF. And so the thought is that more of the neuroblasts survive and differentiate better. So they make better connections because BDNF does that too. So plasticity is increased with BDNF.
So you said something that really interested me about the nature of neurogenesis and the caudate. The caudate, the head of the caudate in particular, you know, first of all, it's misnamed. It ought to be the tail of the amygdala. Second, it's got bidirectional connections to the frontal lobe, which is very unusual in the striatum. And third, it's got its own vascular supply. The recurrent artery of Huebner supplies it. And it strikes me that all of those unusual anatomic features may have something to do with the how or the why of why this region has cellular turnover much more so than elsewhere in the striatum.
Yeah,
Any thoughts about that?
That's an excellent, did the medical students, residents, did you hear? Huber? Very important, right? So
the residents have heard it time and time again.
but this is little blood vessel, and you kind of think, well, why should I study it? The answer. So vasculature, and I don't know the answer, but the vasculature is extremely important for neurogenic niches. So the subventricular zone, if you look at it, it is also fed from the cortex, a very, very long penetrating arteries that go down, and in the mouse at least. And the vascularity of the subventricular zone is much higher than the rest of the brain. So the vasculature, through release of growth factors, stimulate and maintain stem cells. So yes, there may be something unique to that region. The same may be true for the third ventricle. Around the third ventricle, there's neurogenesis happening there, also quite vascularized, as you all know. So yes, this may sort of specify where one has neurogenesis. Any other questions? And I don't know the answer to ketamine. But I'm sure that it does something to neurogenesis. Well, thank you for coming to my seminar.